What up, Mr. Jennings here. Today, here on Big Orange Biology, we're going to talk to you about enzymatic functions. Understanding how enzymes function properly and the things that can affect their function is a critical understanding for the AP exam. Majority of the AP questions that deal with this unit largely lie within the enzymatic function section. Understanding what factors will turn an enzyme on and off or cause an enzyme to change its function slightly is very, very important, and something that we're going to spend a little bit more time on. Now, here on the screen, as you see here, are several factors that affect enzymatic function, namely enzymatic concentration, substrate concentration, temperature, pH, salinity, activators, and inhibitors. All these are various factors that we'll try to simulate the best we can in our classroom and in your investigative studies. Now, factors affecting enzymatic function, there are several of them out there. The first one being enzyme concentration. Now, generally the rule is as the number of enzyme goes up, that's what the arrow here represents, the number of enzymes, as the amount of enzymes go up, so will the reaction rate. Now, usually that means more enzymes equal more frequently colliding with substrate. So while the enzyme itself helps facilitate the collision process that is necessary for reactions to take place, the enzyme still needs to hit the substrate itself. So to help carry this out, you need to have more enzymes. So the more you have, the more reactions you're going to get. Now, eventually, though, the reaction will level off because, well, substrate's a limiting factor. You can only do as much reaction as you have substrate, and over time, you're eventually going to find that more enzymes doesn't equal anything because eventually your enzymes are going to run out of substrates to bind to. Now, substrate concentration will also affect reaction rate. So the same deal here, the more substrate you have, the more likely you're going to collide with the enzyme itself. And like before, the reaction rate will level off. Typically, all enzymes have an active site will be engaged, and that means the enzymes are going to be saturated. There's going to be so much substrate that all the enzymes are doing as best they can. They just cannot keep up with the substrate, and this is what we call a maximum rate of reaction. Notice the reaction rate curve here is this slide and the previous one. They both kind of level off once they hit that limiting factor, which is either the number of enzymes total or the number of substrate. Now, temperature. Temperature affects enzyme much like it affects any protein, because proteins, are highly subjected to their shape. If you change the temperature, you change the amount of each atom and molecule. That means it's going to change its shape slightly. So typically, most enzymes operate under what's called optimum temperature. This is where the greatest number of molecular collisions will happen. In the human body, that's about 35 to 40 degrees Celsius, which is 37 degrees is our human body temperature, or 98.6 degrees Fahrenheit for our non-scientific unit friends. I know Kelvin's preferred, but hey, come on, scientifically speaking, we like to use Celsius here. Now, knowing this, that 37 degrees is the optimal range, you can see that most of the reactions that take place between 35 and 40, 37 is a pretty good halfway point, and that's where we see it. Now, heat beyond maximum temperature will actually increase the energy level of most molecules. Problem is, that will speed up the reaction to an extent, but that ends up creating a problem where the bonds within the enzyme itself will start being disrupted and kind of falling apart. Remember, hydrogen and ionic bonds are kind of weak. And the more energy you have in these molecules, the more likely they are to fall apart. And the enzyme and substrate itself is held together by hydrogen and ionic bonds. If they can't hold it together, the reaction just won't take place and the substrate will fall off the enzyme. Now, denaturation is caused whenever you lose any kind of three-dimensional shape, primarily the tertiary structure. You want to note that. And the same goes here for cold. So warm makes particles move faster and things tend to fall apart. Cold actually causes the reaction to move slower. And because they're slower, there's less collisions happening between enzyme and substrate. Nothing necessarily affecting the enzymatic shape. So the enzyme's losing some shape, but most of it is going to be mainly because the particles ain't hitting fast enough for the reaction to happen. Now, different enzymes function in different environments because of fitness. Every organism has a different fitness relative to its environment, and certain organisms like humans, we have enzymes that work great at 37 degrees Celsius. Meanwhile, bacteria that live in hot springs like geysers, it's not normal. So 37 degrees is pretty cold for them. 70 degrees Celsius, which is 30 degrees shy of boiling, is pretty normal for them. So they're going to have enzymes that work great around those conditions. And we actually, as humans, take advantage of these enzymes found here in these hot springs, and we can do some really crazy genetic analysis with it. Things that you see in what we're going to talk about in biotech. Now, ectotherms are the cold-blooded organisms. So ectotherms have this unique situation where they need to maintain an internal condition that they have to do it through behavioral adjustments. See, endotherms, like warm-blooded organisms like the bobcat you see here, they keep a consistent temperature, so their enzymes work pretty well. Snakes and other ectotherms like reptiles, they have to do different behavioral and feeding adaptations to adjust to the fact that their enzymes may not, because the temperature is always moving. Factors that affect enzymatic function also include pH, and changing the pH meaning adding or removing hydrogen ions. Now, calculating the actual pH from the logarithmic function is a little bit beyond the scope of this course, though you may be asked to do it. We have not seen very much of that. But you do need to understand the concept of adding hydrogen ions. So the more hydrogen ions you introduce, the more pH you gain. Or lower the pH becomes. It becomes more acidic, I should say. The more hydrogen you have, the more acidic it becomes. So things like hydrochloric acid have a high concentration of hydrogen ions. What this does is it disrupts the bonds and changes the 3D shape because there's an attraction between the charged amino acids and the ions. This will affect the secondary and tertiary structure and also denatures the protein. There is such things as optimal pH. Humans, the optimal pH is between 6 to 8. And ultimately, though, depends on localized conditions. So the things that are found in your stomach, like pepsin, pH runs around about 2 or 3 there. So their enzymes tend to work best there. But tryptophan, which is found in the small intestine, which is fairly basic because of high concentrations of ammonia or ammonia derivatives, you're going to see pH of 8. So you're going to work best in a basic condition. Meanwhile, your blood works at around neutral. Other factors such as salinity can add or remove cations or anions. Basically, you add more positive and negative ions. Same situation that you would see with acidity. More hydrogen ions floating around with acidity. Same deal going on with salt. So the more ions you have floating around due to the salt, the more disruption you're going to get the 3D shape, meaning tertiary and secondary structures are going to be denaturing and causes all sorts of disruptions. Enzymes are very intolerant to extreme salinity. As you see there, Dead Sea is called Dead Sea for a reason. Typically, salt is a really good preservative because very few things live in high salt environments. And those that do have to have high adaptations. Now, there's things that help enzymes. So what we just got done speaking about was factors that can limit enzymes or control them. These are factors that can help them. The big ones being activators. There's things like cofactors, which are non-protein, non-organic molecules, things like ions like magnesium, potassium, calcium, zinc, iron, and copper. And what's going to happen is these will bind to the enzyme molecule, like say iron in your blood, found in hemoglobin, and that's going to help facilitate the reaction. It's going to help the reaction take place much more efficiently. And coenzymes, which are non-protein organic molecules that will bind temporarily to the site, things like vitamins. These are what we call our NAD, our electron carriers. We'll talk more about these in the next unit, but these are examples of vitamins that are found as coenzymes. And we'll talk more about those later. Now, for every cofactor and all this stuff, there's things like inhibitors. So these are factors that reduce enzymatic activity. Things like competitive inhibition, non-competitive inhibition, or irreversible inhibition, and feedback inhibition. Now, inhibitor or substrate will compete in competitive inhibition for the active site. Basically, something can actually go in there and it sort of fits like the substrate. It looks like the substrate enough that it can bind to the enzyme and the active site, effectively blocking the substrate. Things like penicillin do this. Disulfiram also helps treat things like chronic alcoholism, basically blocks the enzyme breaking down alcohol, and that creates some situations where, Now, all this does is, it helps overcome, it can be overcome by increasing substrate concentrations. Basically, if you have an inhibitor situation, just flood it with more substrate. Eventually, the substrate will win. It will slow it down. Now, non-competitive inhibitors will bind to an off-site thing called allosteric site. And that's going to bind to an area away from the active site, but when it does so, it causes the enzyme to change its shape, changes its conformation. Because of that, the active site is no longer the same shape, and it basically helps block it. Anti-cancer drugs and cyanide poisoning are great examples of this, and we'll examine cyanide poisoning. Now, inhibitors can also permanently bind to an enzyme. Those can be like competitors or allosteric. Things like nerve gas, sarin, insecticides. These all bind to the enzymes, basically prohibit the breakdown of stuff like found in neurotransmitters, all sorts of nasty things. And there could be also allosteric regulation in general. These are conformational changes by regulatory molecules. These can be inhibitors and activators, but they take advantage of that allosteric site, and they can turn on and off various enzymes as needed. This creates eventually what's called a metabolic pathway. Chemical reactions are going to control how life is organized. And what it does is allows the chemical reaction to be divided into many small steps, which helps increase the efficiency. It's evidence for evolution. It's an artifact left over from all the various evolutionary processes. But all these little sub-steps actually help regulate the process because it gives it more sites of regulation. And since you have more sites to regulate, it creates efficiency and creates a lot of redundancy processes in general. Now, feedback inhibition is basically a regulation and coordination of the product. Basically, various steps in the pathway can be used as a regulator. So the final product is the inhibitor of an earlier step. Often it's usually allosteric inhibition, but it can also be straight competition. This is called feedback inhibition, though. It's technically a negative feedback loop. It basically prevents the unnecessary accumulation of product. It basically keeps you from wasting all your resources. And we're going to examine this in greater detail in 4 and this is an example of that, by the way. This is the synthesis of isolene, the amino acid, from the amino acid threonine there. Isolene becomes an allosteric inhibitor, basically preventing the degradation of threonine. Now, once isolene is consumed, degradation of threonine is going to resume as well. So as you consume the inhibitor goes away and the process begins again until it reaches a certain point. It stops the process. Now, some substrate can act as an activator. Basically, the substrate causes a conformational change in an enzyme called the induced fit model, as we've discussed before. And what that does is favors the binding to a second site and makes the enzyme more reactive and overall effective. Things like hemoglobin take advantage of these cooperativity, basically by working together, and what it does is allows the hemoglobin to bind a lot more efficiently. So in conclusion, a bunch of these processes that we see in enzymes all come down to its structure determining its function. At this point, I know saying structure determines function is repeated ad nauseum, but it is never more true than now, especially in regards to enzymes. Enzymes are really determined by how well its active site is shaped. It's highly specific. And we just listed a variety of different factors that all change the active site's location, or change how well the enzyme may even collide with the substrate. While the substrate itself, the need for the substrate to collide is gone down a lot when the enzyme's present. The enzyme's still got to be able to, attach itself to the substrate in the first place. So reducing, collision rates or increasing collision rates can all have a factor here. So what you really need to take away and make sure you understand from this is all these different factors that we can use to regulate enzymes. This is not the only time we're going to speak of this. We'll be speaking of this throughout the remainder of this unit, and especially throughout units 4, 5, and 6, especially 4 and 6, where we spend so much time talking about gene expression and how cells are signaled. That said, I hope you all had a great day. Thank you for watching and listening. I much appreciate it. Have a nice one.